Sorry about that. All right, so the purpose of the standards working group is to facilitate data sharing and interoperability among analysis tools within the AIRR-seq field through machine-readable standards for sample metadata, sequencing data, and some basic data processing and analysis steps, some of which you heard Brian mention earlier. Okay, next. So we generate a few different products. The primary thing we generate is these data schemas, and those are open source and machine readable and available on our GitHub site. We also build reference API, R, and Python packages to work with these schemas. And we also maintain the documentation site for our products, as well as the products of a few other working groups, as well as lists of compliant tools, such as those that have been stamped by the Software Workings Group. So what we have engaged in the last cycle is we went through, first and foremost, a merger of the prior data representation working group and the minimal standards working group and a component of the common repository working group. And this let us consolidate all of our standards efforts into one group. That seems to be going very well and has been a lot more efficient. We made various process improvements to how we develop things on GitHub. I don't think we need to go into too much detail on that. It's under the hood stuff. We've done 2 releases. We did a major release, version 1.3 of the standards, as well as a point release with some fixes. The schema that we have in documentation that we have developed in that cycle is that, as was mentioned by William and Corey earlier, we have developed these experimental germline database schema in collaboration with GLDB, mostly the work of William, and that includes provisional support in the R and Python reference libraries. We have developed an experimental single cell schema. We have developed an experimental clonal lineage schema, and this includes a few different subcomponents, as well as this receptor schema, which is primarily a protein-based schema for paired chains, antibody affinity, and external links to things like IADB. We've gone through some cleanup of the documentation, and we have a rough draft worked out for a file manifest, which is a way to inventory files on a file system that are all part of one package, as well as ways to aggregate multiple repertoires. So the progress that we've made on standards development has been pretty consistent, and we're currently up to 23 tools that we are aware of that support the standard. I actually think this list is a little short. But so far so good. Sort of major packages that have come in the last cycle is Cell Ranger and IMGT Hive Quest. We're continuing to work on documentation. We'll continue to refine this. As I said before, it includes a variety of different documentation for both the work of the minimal standards and data representation groups, which have now been consolidated into one effort. Public data repository submission guidelines, software guidelines, which was mentioned by the Software Working Group as well, ontologies and vocabularies, as well as that list of tools that are compliant. If you have any documentation needs for your working group, please reach out to us and we will help get you on the site. So moving forward, what we're going to do is that we have a lot of schema that have been developed in an quote unquote experimental state. And an experimental state is an official release state, but it has not been finalized and is subject to backwards compatibility breaks. So a big effort that we're going to do moving forward through this cycle is we want to refine those schemas that we already have as opposed to develop new ones and get those into production. We're going to continue to outreach as much as we can. And it's probably about time we put together a manuscript because we've made a lot of progress since 2017 and 2018 when the original manuscripts were published. Our release targets, this is something that was also mentioned during Common Repository, is we're looking to get version 1.4 out shortly after this meeting. That will unlock version 1.2 of the ADC API, which will include pointers to those. And this is going to include that germline database schema, single cell schema, receptor schema, as well as some corrections for new technology, particularly in the space of abundance fields. And then we're also going to abandon Python 2 support a little earlier than we'd hoped because development was slow and that's been end of life for a while. And then we have a few minor improvements along the way in various field names. All right, so later in the year, we're targeting version 2.0. And the major thing about version 2.0 is this will hopefully be where we've taken all of those things, we've refined them, we've put them into production quality, including some experimental release of the manifest, repertoire grouping, and some issues related to persistent identifiers that we're hoping to clarify. There will also be some minor backwards incompatible breaks, which is why it has to be version 2. Finally, I'd like to list all of our members that have contributed, insight or code or anything during this last cycle. And going forward for this next and final cycle, Christian and I are volunteering to be co-leaders. So with that, we'll close out. Please join us. Everybody has this page. Here's the links to our documentation site, our GitHub, and where you can get in touch with us. Questions? Good, perfectly clear.